In mini project, we are group 12 in subculture for routing. I will introduce our team member: Siren, Natasha, Pindi, Muhammad Sepila, Nur Shaira Pindi, Ahmad Hamga, Muhammad Lufan bin Muhammad Kairi, Muhammad Kamal Hamdi bin Nazan, and also me, Kechunting. In objective, the routing subculture is a technique that used in orchid propagation to produce new plants from the existing one. The purpose of subculture for the routing is change the oxynidium that only with shoots from media that without hormone to media that contain active charcoal for the root induction. Root subculture in this context refers to the specific set of techniques and conditions employed to encourage the development of root in orchid plant. This is because the roots are important to it for nutrient absorption.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Lufan bin MD Khairi. Nombor matrik saya F2 1B 1381. Okay, first introduction. Subculture in this context refer to the specific set of technique and condition employed to encourage the development of root in orchid plants. The importance of subculture for root development of Cymbidium finlaysonianum are to root induction. The objective of subculture for rooting Cymbidium finlaysonianum are propagation, mass production, and also rapid growth. The primary objective is to produce new plants from the parent plant. Media in subculture of root providing the right condition for the development of root to further nutrient absorption. Subculture can be used for mass production of Cymbidium finlaysonianum plants by creating multiple plants through the rooting process. Nurseries and orchid enthusiasts can increase the availability of this orchid from commercial or personal purposes. Rooting subculture provide a controlled environment with the right nutrient media and condition to promote rapid root development. That's all from me. Thank you.
Hi, my name is Nur Shaira binti Amamka. My matric number F21B1179. So I'm gonna talk about materials and method. So first material that we use is parent plant material is Cymbidium finlaysonianum, the microshoot that ready from lab tissue culture. So the second material is sterilization agents is Clorox 70% of ethanol spray. And the next one, culture medium MS is Murashik and Skog. The medium at which active charcoal for promoting root formation. Other than that, tools we use is scalpels and blades for taking the shoot and forceps for handling the shoot. And the last one, flame source for sterilizing tools like oil lamp. Okay, about the method. We sterilize the forceps and scalpels by using oil lamp, and then we uncap the jar, carefully remove out the microshoot from the jar, and we separate the microshoot into individual using fossil to transfer them into root media is MS, Murashik and Scope. Medium add rich active charcoal. And then we recap the and seal the cap with parafilm to make sure the culture will fine. And we label the culture with type of medium and write the name, group, date and plant species. And the second last, keep the culture in the control growth room. The temperature is must above 25 Celsius, 60-hour photo period. And then the last one, observe the culture ever to 3 days and record the observation. Thank you.
Hi, my name is Zali Natasha binti Muhammad Zafina. My metric number F21B1273. Okay, I will discuss about the result and discussion. Okay, the result in the experiment of subculture for rooting is 36 of jar Cymbidium has been used to subculture, and only 3 jars of 36 jar that contaminate from fungus. We also estimate the successful rate height due to 91%, while we found root growth to be very effective because they are began to grow after 1 week of subculture. So next, the discussion of this result. The result of the subculture for rooting experiment involving 36 jars of Cymbidium are promising, with a high success rate of 91%. This indicates a robust and effective subculture process. The minimal contaminate rate with only 3 out of the 36 jars affected by fungus highlight the overall sterility and precision maintained during the subculture procedure. The success rate of 91% suggests that the subculture condition, including the medium composition, environmental factors, and handling technique, are conducive to the healthy growth and development of Cymbidium. This high success rate is a positive outcome, as it indicates the reliability and efficiency of the subculture method employed in promoting rooting. And from the observation that roots begin to emerge after just 1 week of subculture further emphasizes the rapid response of Cymbidium to the provided condition. This quick initiation of root growth is crucial for the overall success of the tissue culture process, as it signifies the plant ability to adapt and thrive in the new environment. And lastly, in conclusion, the subculture for rooting experiment demonstrates a commendable success rate, minimal contamination issue, and prompt initiation of growth of root. This outcome suggests that the employed subculture protocol is effective for Cymbidium, contributing to the advancement of tissue culture technique for this plant species. That's all from me. Thank you.
My name is Muhammad Kamal Hamdi bin Azan. My metric number is F21A1141, and I will talk about conclusion. Subculture for rooting of Cymbidium phenolothonianum is a practical method used to reproduce and propagate this orchid species. By employing techniques like tissue culture of pseudobulb divisions, it enables the generation of new plants in a controlled environment. This approach is valuable for conservation efforts, ensuring the preservation and wider availability of Cymbidium phenolothonianum. While it may depend precision and attention, subculture for rooting serves as an effective means for enthusiasts and experts to multiply and share this orchid, contributing to its sustained existence and enjoyment. That's all. Thank you.
Recommendation: the media must use MS, Murashi and Scope medium that act with active charcoal for roots development and enough nutrient for culture to grow. This is because MS media contain macronutrients, micronutrients, vitamins, and serum that suitable to Oxynidium to grow. In this stage in tissue culture, the procedure and environment must be in hygiene to avoid contamination of fungus and bacteria. The temperature must in above 25 Celsius 16 hours photo period to give the culture to grow by photosynthesis.